Hello, my name is Adina Mangubat, and I'm the CEO of Spiral Genetics. And this is an actual pitch deck that I've used to raise money, so to give you kind of a feel for what it's like. It doesn't have all of the financial data, because we can't include that for public things, but hopefully it'll give you a feel. So I started Spiral Genetics because I realized at a pretty young age that we're actually all affected by disease. And this really came from me in the form of my grandfather. He was diagnosed with lung cancer when I was 15. And what was really striking to me at that age was that my dad is a doctor, and that didn't matter. And we were in Seattle, home of some of the best cancer treatment centers in the world, and it still didn't matter. In the end, nobody actually knew what to give him that would make the difference to save his life. And it was very interesting because that was the first instance of it, but now my grandmother has Alzheimer's. And so it just became very, very clear to me that we're all affected by disease, and it's just getting worse because the population of the world is aging. And so if we're not, taken out by something like getting hit by a bus, right, ultimately, we're all going to be affected by disease, whether it's ourselves or someone that we actually care a lot about. And so I was talking with my co-founder, Becky Dries, who's a molecular biologist PhD out of Berkeley, and she said to me, look, if we're going to have any shot at understanding any of these diseases and having cures that are actually going to work, we have to understand what is happening in the fundamental blueprint of who we are, what's happening in our DNA, what's happening in our RNA. And so that's why we created Spiral Genetics. Now, the idea of personalized medicine is not new, but I would argue that a lot of it has failed to date because the studies that people have been running are way, way too small. And you can actually see this in a lot of the different research studies where you're having groups of a couple of hundred people, and you're trying to compare them and figure out which genetic variations are the ones that are important for disease. But if you compare you, any of you, and me, assuming we don't have a great-grandmother we don't know about, we're going to actually differ by 3 million genetic variations. And so which ones out of those 3 million are the ones that increase my family's risk for diabetes? How do you pick that out of comparing a couple hundred people? You just can't. The statistics don't work. And historically, the reason why these studies have been so small is because the cost has been so high, and the throughput for doing DNA sequencing has been so low. But that has recently changed with the launch of the $1,000 Genome Project. And so now you actually have 500,000 people that were sequenced last year, and there's supposed to be several million this year, which is great, but the big challenge that people are all now facing is the data analysis portion. All of the software that they have for actually extracting the meaning out of all of this data is insufficient. And the big issues that they deal with are really data management, because these are very large files, 100 gigabytes per person. And so if you start multiplying that out by 500,000 people, you can kind of get an idea of how massive a data problem that really is. The data analysis techniques are really slow, so it only really allows for one-time analysis. And there's no scalable way of comparing large groups of people. They're built for comparing hundreds of people, not hundreds of thousands of people. And so that's what Spiral Genetics does. We figured out a way to take all of this data, compress it down dramatically, taking it from 100 gigabytes down to 3 gigabytes per person, which means that you can stuff it into RAM memory. So any of you computer scientist folks out there, that's kind of how we get the rapid speed in analysis. And you can quickly add people to any study. So adding new individuals is not a massive reanalysis. It's very, very simple. And you can actually compare hundreds of thousands of people simultaneously. So you can actually get answers in weeks rather than months if you actually have the data. Now, all of this has actually been validated by a number of scientific collaborators, the biggest one of which is Baylor College of Medicine, and they say that what we're doing is absolutely crucial for all of the large-scale studies that they're doing. And this is a big deal because it actually provides a barrier to entry to this industry. In order to actually sell into these groups, you actually need scientific credibility because no one will take you seriously. So it's not like somebody can just waltz in and have a new solution instantly. The go-to-market is very simple, really. We sell to large sequencing instrument centers, so people that have large amounts of sequencing capacity. We sell them first on the data compression aspect because that saves them money up front. And then we upsell them on the data analysis and comparison as they get into their more complex questions that they want to ask. Today, we have basically 9 major collaborations in a huge different array of disease areas. We're working with 3 different countries on their large sequencing projects, and we're working with Baylor College of Medicine on some of their large projects as well. So very, very powerful technology. The business model is also very simple. We sell a site license to these institutions, and it's based on the amount of sequencing capacity that they have. So it's about $100 per genome. And if you look at the overall market size, you can estimate that very easily by looking at how many genomes there are predicted to be done per year. And by 2020, we're looking at 20 million, according to Illumina. So it's a $2 billion market size. In terms of potential competitors, there are people that are doing DNA analysis and doing bioinformatics, but they're really all built for the analysis of individual genomes or small groups of individuals. And they can't really compare people at population scale. They cannot find a lot of different types of genetic variations that are really important for disease, and specifically for cancer. The team that backs up this, I've been very, very fortunate to put together. Jeremy Bristol is our Chief Technology Officer, a brilliant computer scientist, and works very closely with Niranjan Shekhar, who's our PhD statistical geneticist and has 15 years of genomics experience in the field, along with Stephen Weil, who's our VP of Business Development, a Master's of Science, a Master's in Computer Science, as well as many, many years in business development, which makes an amazing combination when you're selling into this industry, along with our advisory boards as well. And I always pitch, whenever I pitch coach students, I always ask them to make an ask at the end, and so I'm not asking you for money, but we are hiring. So if you have any folks that are interested in a bioinformatics position, do let me know. Thank you.
I don't know how many of you guys have thought of this, but my background is actually not in biology, it's in computer science and physics, and computer security in particular. And I got to thinking that the body is driven a lot more by information than it is by chemistry. And I mean, obviously chemistry is the substrate, but if you consider, like, without the information contained in your DNA, you're a few bucks worth of chemicals sitting in a chair right now. With information, your life. I mean, you eat, sleep, breathe, reproduce. And so I got thinking, why is it that we don't treat diseases with information instead of chemistry? And I guess, the obvious answer is cells don't have USB ports. You can't just, like, upload new treatments for them. But I'd say pause for a second and consider how different medicine would look if we had an app store for the human body. If you could treat a disease by uploading a new program to your body. And as it stands right now, some of the most effective, powerful treatments we have are biologics, things grown in living cells. And these are great. I mean, they're kind of the best and the worst in that they tend to be very targeted, very powerful, but they're also very hard to produce. They tend to be very expensive. They're not stable in shipment. Often have short half-lives, and you often have to inject them. And so we weren't particularly happy with the status quo here, and so we decided that we are going to try to build the USB port. We're trying to build the App Store instead. And so basically, instead of making a treatment outside the body and injecting it, we'll take out some of your cells, B cells, an immune cell that normally secretes antibodies, and we'll give it instructions to produce a therapeutic protein inside your own body. And these are really ideal vehicles for this because they're probably one of the most prolific protein producers in the entire body. They're dedicated factories, and they can have a very long life. The half-life of a long-lived plasma cell, in fact, is about 17 years in the published literature. So if you take the blood from an 80-year-old, you can still find antibodies against smallpox in circulation. And they're being produced by the cells that were generated all the way back when they got challenged by that vaccine or the virus. And I guess a perfectly awful example of why this is useful, and our first indication actually, is a disease called MPS I. So it's a rare genetic disease in which patients can't produce an enzyme they need to break down long sugar chains in the lysosomes of their cells. And untreated, it's lethal by, I think, age 10 or so. And typically now they'll, at least in the US, 1.5-year-old kid will undergo myeloablative chemo and they get a stem cell transplant, and it's brutal. And the ones that survive that make it into their 20s, but they're in pretty bad shape. And you can also make the enzyme recombinantly and inject it. And this is an approved product made called Eldurazyme by BioMarin and Genzyme, or now Sanofi. And, but it costs at least $250,000 a year, and you have to get a 3-hour-long infusion once a week at a hospital for this. And the half-life of the enzyme is so short, it's about an hour and a half or so in plasma, that it's effectively gone by the time they get home. And so we got thinking, well, what if we made your body produce it 24/7? I mean, you think of how that's going to alter the PK of the drug, if nothing else. Like, instead of having a big spike and a crash, you have stable production for potentially years. So the way it penetrates tissue is different. The side effects from the high dose go away effectively. Like, your area under the curve expands even though the concentration goes down. And this, you know, it's a relatively small drug by biotech standards. It makes a little over $200 million last year. But the beauty of a platform like this is you can basically make a new treatment simply by changing the DNA. And so we think, in addition to being able to do some good in treating out nasty disease, it also really shows the power of the platform. Before I go too far, I'll give you a quick idea of how this treatment actually works. And it's not too dissimilar from the CAR T-cell transplants, the cancer immunotherapies we have today. Basically, the patient goes in for a blood draw or apheresis, typically in our case, and the B cells are isolated with immunomagnetic beads, and we modify their DNA. And we actually used to do this with the virus, and now we use something called a Sleeping Beauty transposons, non-viral vector. It's dirt cheap. Like when we're using the virus, I think we budgeted $1.3 million for virus alone in our phase 1 clinical trial. Now it's a few $100s of the DNA. It's quite cheap. So we expand the cells in vitro, and we can do this up to 10,000-fold. We're really, really good at culturing B cells. It's our core technology. And as we do that, we mature them into plasmablasts, which are the immediate precursor to a plasma cell. And the great thing about these are, when you put them back, they'll migrate on their own to survival niches, primarily in the bone marrow and spleen. They have a chemokine homing receptor that guides them, so you don't have to do any conditioning or anything like this. You just inject them, and they go where they're supposed to. And like I said, if they get there, they're expected to survive for many years. So far, we've been doing this. It's kind of interesting in that our treatment was human cells from day one. So you know, a lot of times you think of a typical drug development where you screen a bunch of compounds, you put them in a petri dish, and a mouse, a monkey, and then a human. Ours is human cells forever. And so that actually complicated the preclinical work. So we can put these in immunocompromised mice, though, and show that they do migrate properly. The chemokine homing receptors are cross-reactive. Right now, we can put them into mice and actually take a MPS 1 mouse, a model of the disease, and bring the levels of IDOA in circulation up to 50% of healthy endogenous levels with one treatment. And it's awesome because it's thought that getting to 10% is curative. And so now you have a tool to make this incredibly expensive, cumbersome treatment in your body for basically free for life. And so mice, they don't survive super well, but mice don't live 17 years anyway. So this is what we've got so far, and we are moving forward to the FDA now. I'll just touch on this briefly for time, but we have the ability to make pretty much anything you'd like. And I think some of the real killer apps for this are things like age-related sarcopenia, doing things like building muscle with old age, and recreating effectively the biochemical environment of youth as you get older. So like far larger applications than what we're looking at for rare diseases. We recently purchased a company that developed the Sleeping Beauty transposon, and we've done our pre-IND hearing with the FDA, and our goal is to actually apply to begin human trials by the end of Q3 this year and start human trials next year. Just in the interest of time, I won't go through the team very much, but I'll say the core technology came out of a Nobel laureate's lab at Caltech, and we made off with the inventors of the core vector technology, who've been doing gene therapy since basically before I was born. And we have raised $5 million to date, and we're after a $10 million Series B right now.
Thank you very much.
Thank you. Well, as a trained immunologist from that other institute called Oxford, from the Dunn School of Pathology where penicillin was first purified, I feel very comfortable with the disease focus. But I think we have to remember that we got to feed, fuel, and provide clothing for a growing population. So my company focuses on plantation forestry and biotech of plantation forestry. If we look at the driver for why biotech in this field, essentially the data here comes out of Brazil, which is one of the largest plantation forestry countries in the world, has 5 million hectares of eucalyptus. And you can see here that basically the eucalyptus yield back in the 70s was 21 cubic meters per hectare per year. And through good silviculture practice, conventional breeding, this has been more than doubled over the 40-year period, but it's reached a plateau. And conventional breeding today manages to raise yields by about 1% per 12-year breeding cycle. We need biotech to push this up. And we've developed technologies where we can enhance yield, and we're increasingly focusing on technologies where we can protect yield. Because with climate change, globalization, travel, we're seeing an increasing profile of novel diseases and pests which are attacking and detracting from yield. So if we look at the technology evolution in the GM plant field, the first generation dealt with herbicides, herbicide resistance, insect resistance, separate traits in separate crops. Second generation, or 1.5 generation 1.5, stacked these traits. But now we need to move to a next generation of technologies where we can regulate output pathways, yield, nitrogen use, abiotic stress, fiber properties, quality traits. Some of these have existed for years, but part, and this was mentioned earlier today, part of the success in this field is regulatory, and a major issue in this field is public acceptance. And I really identify with what Sir Greg said earlier. What we put into our body for acute issues and what we prepare to do for chronic issues. And there's just a major factor in this, and I can speak for days because I've been at the forefront of genetically modified trees. People run after organic food. Organic food uses 20% more resource to produce the same amount of yield. Is that moral in a world where we need to be producing more from less? We'll talk more about that tomorrow morning as well. The vision of my company is to be a world leader in developing sustainable genetic solutions for the plantation forestry industry. Why is the plantation forestry industry important? 7% of the world's forests today are renewable plantations. They produce 50% of our industrial wood. If we detract from plantation forestry, we go back to extraction from natural forest. Natural forests are the lungs of the world. We need to preserve that resource. The sectors we deal with include fiber production, pulp for the pulp and paper industry. Although writing paper is decreasing as a commodity today, the world is using more and more tissue. As people develop, tissue paper has become a bulk thing. Paper growth is still going. But fiber can be used as a renewable source of chemicals. Again, talk more about that tomorrow. But essentially, 66% of our chemicals today come from fossil fuels, non-renewable. The platforms we deal with are yield and processability enhancement, yield protection. The crops we deal with, eucalyptus, poplar, and desert species. We're very involved in domesticating desert species, especially in China, where 17% of the landmass is desert and growing. Our markets, Brazil, China, the USA. The history of our company is a business school case study. We started off angel funded. We went through venture funding, up rounds, down rounds, play-to-play, and near-death rounds. I worked for 2 years without a salary. Essentially, we moved into the agricultural biotech field in around about 2000, 2001. In 2006, I merged the company, which was then called CBD Technologies, Cellulose Binding Domain Technologies, into a small startup that had come from Purdue. We were on the London market for 4 years. In 2010, we were bought out by a large Brazilian company. And a key milestone in 2015, we got approval for the first commercial genetically modified eucalyptus with a 20% yield increase. How did this technology start? We were actually looking to try and inhibit cellulose production. We had these molecules called cellulose binding domains, which we'd isolated from bacteria. We incubated them with small plants. We were looking at trying to automate deciduous fruit thinning, where fruits are taken off the trees to allow the other fruits to grow better. We incubated these CBD molecules with seedlings, and we got a strange dose curve that at low dose, we actually got a stimulation of growth, the second plant on the left. Unraveling this mechanism took us to cellulose biosynthesis. Cellulose biosynthesis is initiated in the plasma membrane of the plant cell, where cellulose synthase rosettes produce cellulose microfibrils, which then go and aggregate into crystalline cellulose. We found that these cellulose binding domain proteins could actually act as biolubricants and could uncouple the rate-limiting steps of this crystallization process. It was a protein. It had a gene. So we took that gene, we targeted to the cell wall with a cell wall-specific sequence. We introduced these into plant cells, and we found we could modify the plant cell wall. By modifying the plant cell wall, we could accelerate the growth of the plant. This was actually not the first protein we took to regulatory. In this process, we discovered a range of proteins in the plant cell wall, including an beta-1,4 endoglucanase, which was involved in editing cellulose. And that is the first protein that we took through to regulatory approval. This is the notice from the Brazilian government. We went through, when I talk about public acceptance, in this process of getting regulatory approval in Brazil, we had a facility invasion. 500 activists invaded our facility, destroyed greenhouses, destroyed planting material. Quite an experience to go through. On the same day, they invaded the Brazilian government's regulatory commission, disrupted proceedings. I had to go back a month later from my Israeli background, organize a security process in Brasilia to allow a government commission to sit and to actually take approval. We have an increasing pipeline of products, including yield enhancement through to second generation yield, to yield protection, a large range of public-private partnerships. I'm going to end on that note.
Thank you very much.
So, disclaimer, today is actually my first day at working for Agenus. So I'm actually picturing when I haven't actually met anyone from Agenus yet, apart from who I met in my interview. However, I'm going to, luckily, Sophie had given a good introduction about immuno-oncology, and Greg had given a good introduction about antibodies, so I'm hoping that my work is half there already. So, if you look at cancer, and these are the current strategies for combating cancer, really there are 4 ways that you can try and tackle cancer. Either by sort of chemotherapy, and that's using small molecules, or even better now that we can actually target antibodies attached to small molecules, cytotoxic drugs, to try and attack cells, tumour cells. There are lots of small molecules out there that actually go into the cell and actually can start to target cell pathways that you can kill the cell and the tumour cell with. However, you've got 2 new areas here that have just come out, which is all about immunotherapy, where you're actually using a smarter way to try and kill a tumour cell. Instead of killing a cell, you actually reprime the immune system to kill itself, essentially, to kill its own tumour cell. And this is the way that the strategy that Agenus is taking. It's really based on 2 drugs that have come out recently, Keytruda and Yervoy. Keytruda is an anti-PD-1 antibody, which you can see here, and it breaks the PD-1-PD-L1 interaction. And then here you've got Yervoy, which is an anti-CTLA-4 antibody, which breaks the interaction between CTLA-4 and its antigen B7. So what happens is that your tumour expresses these immune checkpoint inhibitors, immune checkpoint antigens, and that's what your body thinks is a self-antigen, therefore, and it doesn't attack it. But if you break these interactions, your immune system goes, oh, this is a foreign entity, and it reprimes your immune system, and it attacks your tumour cell. And this has shown great success so far with these 2 antibodies, where cancers that have never been able to be curable before are finally have high success rates. So at Agenus, the strategy there is that we are an immuno-oncology company. And when you've decided to yourself that I'm going to go for a particular disease area, the strategy that Agenus is taking is that it doesn't matter what the platform is, we are going to be tackling cancer through immunotherapy. And the way that Agenus is doing this is by throwing everything at it. Primarily, it's all about antibody technology. And we'll be using things like Phage display, which is the team that I run here in Cambridge, as well, but with everything else attached to it. So Agenus use something called yeast display, where you can actually express whole antibodies in yeast and make libraries, and then you can find inhibitors for that. There's retrocyte display, which is a form of mammalian display, also again to try and increase your chances of getting a good hit. We do things like FC engineering to generate bispecific antibodies and enhanced antibodies to be able to do this more effectively. We develop immunobiological assays to be able to assess this effectively, and computational biology and bioinformatics to be able to analyze the results that we get. In addition to this, Agenus is also trying to personalize cancer by making personalized tumors, and by two main techniques called prophage and autosynvax. And by this, you take out some of the cells from an individual patient and raise antibodies towards it to make a personalized approach to vaccines, so similar to what Sophie had described earlier. And we think that by doing this, by having a two-pronged, all-pronged approach, we'll be able to successfully combat cancer through an immuno-oncology perspective. I'm going to end it there. I'm going to keep it very top-heavy. These are the senior management team in Agenus who are involved in trying to generate antibodies for immuno-oncology, and the research and development team here, in particular here, Alex Duncan. I'd like to acknowledge him, who's my boss, who is heading up the antibody technology aspect of it. Thank you.
Good morning, everybody. So I have a microphone, but I am quite loud, so I'll perhaps dispense. I am using the microphone, am I? Oh, okay, good. Hopefully I won't deafen you all this morning. So my name is Emma Sceats. I'm CEO of CM Bioinnovations. I only have a small handful of slides to walk you through this morning. So what my company does is we develop human organ-on-chip technologies. And what these are tablet or smartphone-sized devices that allow us to build miniature replicas of human organs in the laboratory.
Can you put your microphone close to your mouth?
Sure.
I'm recording it, so
Okay.
I think it's...
So we're what's known as a tools and toys company. So we provide products and services to pharmaceutical partners based on these organ-on-chip technologies to help pharma companies develop better, safer medicines more efficiently. So why are we doing this? Well, the pharma industry is still very successful despite a lot of gloom in the industry. It made $1.15 trillion globally in 2013, at least according to The Economist. But the issue is one of productivity. It could be a parable for our times, I suppose. The issue is that fewer drugs are costing a lot more money for the industry to produce. And there are a lot of issues that have been pointed to as being causative, and I'm sure you were all busy reading the business pages of the Sunday newspapers yesterday when Luke Johnson, for instance, raised in the back of the Times the fact of short-termism amongst senior executives and shareholders as being one potential cause. But we know that that's an oversimplification. Sophie's spoken broadly about how pharma are trying to approach big problems that will be many years in the making for pharma to bring drugs to market. So I'm going to speak about two issues that really go to the heart of the science of pharmaceutical drug discovery. And the first one here is the use of animals as surrogates for humans during drug development. Obviously, the regulator requires that new medicines be tested in animals prior to clinical trials. But undoubtedly, there have been a number of cases, really too many cases, of where drugs have reached the clinic, having successfully passed through preclinical development, and failed to be either sufficiently safe or effective to be granted approval for use in patients. And it's clear that we have been overlooking the differences between humans and these animal test subjects. So I'll talk about some of the things that we're doing to address that. But a second point goes to the heart of the methods that have been employed in pharmaceutical drug discovery, and especially some of the methods that have gained prominence through an effort which has really accelerated through the 90s and the 2000s to industrialize aspects of drug discovery. And that is, if we think about the sequencing of the human genome, that happened in 2001. Whilst there have been many success stories as a result of understanding the human genetic code at a deeper level, biology and drug discovery is unfortunately a bit more complicated than the interaction of a drug with a single protein target. And so we are thinking about, and I'll show you some of the technologies, approaches where we can take a much more holistic approach to understanding the biology and the interaction of drugs in these complex cellular and tissue-based networks. And I'm going to talk about some of the in vitro models which we combine with omics technologies and with some computational modeling techniques to help us understand some of these more complicated multifactorial diseases that have been spoken about this morning, such as depression, Alzheimer's, metabolic diseases, and obesity. So I'm going to talk about just two examples of technology that we've been working on at CM Bio. And so I'm going to counterintuitively, I'm going to move from right to left here. So on the right-hand side, we have developed at CM Bio, based on technology in-licensed by our company from the Massachusetts Institute of Technology, a liver on a chip model, which we can infect with hepatitis B virus directly from patient samples. So we can take patient serum, inoculate cells in our liver on a chip device, and generate a full viral life cycle infection of hepatitis B in our platform. And the neat thing about this is we can probe in some considerable detail the interaction between the virus and the host platform. And this is a technology that we have worked now with 6 pharmaceutical companies using this technology to help them advance their programs in hepatitis B. And I'd say in hepatitis C, which has really shown the way for some antiviral discovery research in recent times, Hep C is a great example of where the pharmaceutical company shows that it is committed to developing cures for serious diseases, and hopefully the same will happen in hepatitis B. And on the left-hand side here, I'm just showing an image from a demonstration that we provided to the US Department of Defense in February of this year. So the director of the DARPA, a $3 billion a year funded agency in the US, we're currently part of a typo here, $26 million US defense-funded program to develop a human body on a chip. And this is a platform that shows 7-organ interaction. And clearly the type of information we can get out of these devices is so complex that we need to rely upon computational systems biology modeling to help make sense of them. But yeah, just 2 examples, and I'll be here for the rest of the day, and I will be happy to speak to anybody about this in more detail. Thank you.
Thank you. I'd now like to open the session up for questions from the audience. One right at the back.
Okay, so hi, my name is Marcus. I'm with AstraZeneca, and with a business background, I have a question for FuturaGene. And I was reading an article the other week in The Economist about how pulp yield has gone up through innovation. But it also said that now the growth in supply is outpacing the growth in demand for tissue, and that is mainly used with manufactured with pulp. What are your thoughts from a kind of a risk perspective in that innovation has to be applied innovation and has to meet a demand, and when innovation is outpacing demand, then we're basically just flooding with the market with something that's not needed?
Pulp production is outpacing the market, but that doesn't relate to where that pulp is coming from. And the basic thesis that I would put forward is that we need to be producing more efficiently from less forest footprint to produce the pulp that is needed. The pulp market is like most other commodities. We've seen a glut of commodities, and then we've seen a down cycle. There was a massive demand for pulp over the last few years, which drove pulp prices up to about $800 per ton, which drove factories to put on capacity. It still comes back to where do you take that pulp from? Speaking to some of the people here this morning, there are people, for instance, from Indonesia here. The bulk of Indonesian pulp production, which is a major factor on the world market, is coming from unsustainably logged wood and forests that are not replanted. Africa still has major deforestation. So we need to focus down on how much of the commodity we need and how do we produce it.
I mean, I think you make a good point about having a global bioeconomy strategy and the trade-offs between, the various sectors in terms of for food or for pulp in this particular case.
I absolutely agree with that. And the last comment on that is that if you look at forests, and especially plantation forests, this is a huge lignocellulose resource. What we need to be asking ourselves is what products are we making from that resource to come to the global economy,
Very good
bioeconomy.
Point. Another question there?
Hi, my name is Donna, originally from Taiwan and now a first-year MBA student at Duke. I have a question for Matthew. I'm really curious about the business you mentioned, because as far as I know, for example, for MPS 1, how do you see the competition from some mid-sized biotech company or from some large biopharmaceutical companies, for example, like Genzyme or Biomarine? Thank you.
That's a great question. We kind of have a love-hate relationship, I think, with pharma. And when we were starting out, my general impression was they'd be happier if I were found floating face down in the water than bringing this platform forward. I mean, you think about it, we don't compete in a traditional sense. I mean, if we treat you, you're good. That means Biomarine can't steal you back or try to lower their prices and then it's game over. So the way we've interacted with pharma has been interesting, and I'd say right now that the reception is quite warm with these guys. I mean, we approached, interestingly enough, Biomarine when we first started. I said, hey, I've got an idea. I would like to know what I need to show you to convince you this is a good idea, and I will show you all the data, and I'll give you first right of refusal to take it if you like it. And they totally to shut it down. But people aren't doing that anymore. And I was pretty blunt with it in some of these meetings. I'm like, look, this is a weapon. Either you're going to be aiming it or you're going to have it pointed at you. And it's your portfolio that'll be toast. And so that might not seem like the most, say, ecumenical thing you can say in a meeting with a potential partner. But I think that is indeed the case. And beyond that, though, there's a ton of unmet needs. So MPS 1's become a whipping boy for gene therapy because there's lots of things out there. I don't think anyone's looking at building their entire company on it. But it's more showing that it works. But there's just countless diseases you can go after. And like I said, I'm after much bigger fish. Not everyone gets MPS 1 or HIV, but everyone gets old. And I want to be able to try to address these larger cultural issues.
Okay. Any other questions? Yep, let's put one over there and one down the front. Got time for... we'll have two. Yeah, two, but can you make them short and can the answers be succinct?
Hello, my name is Adina from Malaysia. I'm currently working as a credit analyst at Agricultural Bank in Malaysia, at the same time working for my plant biotech startup. So I have a question for FuturaGene. I work on a pulp and paper project when I was 16 years old. So I was like really surprised on how huge water consumption to produce pulp and paper. So I wonder how effective your technology in reducing water consumption to produce high quality paper. Thank you.
Brief.
Yeah, very brief. Second generation technologies we're working on are looking at modifying fiber properties and cellulose lignin interactions, which will allow you to actually produce the pulp from the basic wood far more efficiently and lower consumption. Water recycling technologies are a major factor in terms of putting back clean water into the system. 95% of the water that is used goes back into the system.
Okay. Fine. And last question. Thank you. Can we make it brief?
Hello, I'm Lenia Swenda from Medicines for Africa. I have a question about the transposon technology that you're using. How different is it from the vectored vaccine expression systems, and are there any, what are the regulatory challenges that you anticipate when you're going to the FDA that are specific to using transposons compared to vectored vaccines, which have been used quite a lot already?
It's hard to answer briefly, but in general, the transposon's been used in clinic 3 different times now. It's got an integration profile that is as close to true random as you can get. It's basically regarded to be as safe, or maybe a little safer than lentiviruses. So it is integrating, and we expand the cells in vitro. In the case of Africa, we actually did a Ebola prototype where we're doing in vivo electroporation to make the antibodies for Ebola in muscle. And so that actually is probably of more interest to you. We can talk about that later. But the FDA has looked at this pretty favorably. They're letting us go, at least it appears, into humans with this relatively cleanly.
Well, perhaps you can follow up afterwards.
Yes.
So I'd like to thank our panelists, speakers once more, and I think coffee is available. So can we just thank our speakers?